Welcome students to St. Joseph Open Web-based learning platform So We Learn. Today we are going to discuss with another vascular cryptogram called Rhynia. If you see the systematic position of Rhynia, it belongs to division Xylophyta, class Xylotopsida, order is Xylophytales, family is Rhyniaceae, genus is Rhynia. The fossil plant Rhynia, belonging to order Xylophytes, consists of 9 families and 25 genera, all of which are fossils. They show that they became extinct during the Upper Devonian period, and they lived in the Middle Silurian to Upper Devonian, and they remain to Xylophytes have been found widely scattered regions such as Canada, Scotland, France, USA, and Australia. There are two species of Rhynia have been reported by Kinson and Lang in 1917 and 1921 from an old red sandstone beds of Middle Devonian time in a place Rhynie situated in Scotland, and hence the name Rhynia. Two species of Rhynia are Rhynia major and Rhynia gwanvongani. If you see the morphology of Rhynia, the plant body is herbaceous, where the Rhynia major is 50 centimeter in height and 1.56 to 5 to 6 in diameter, while Rhynia vaughan was only 20 centimeter in height and 1 to 3 in diameter. The plant body was differentiated into subterranean rhizome with an abruptly turned upright photosynthetic aerial shoots. Roots were absent, but at places rhizomes were provided and in tufts of unicellular rhizomes. The aerial shoots were cylindrical and leafless with a tapering dichotomously branch system. In Rhynia major, the aerial shoots were smooth. Rhynia Vaughan was adventitious branches were present on the aerial shoots as well as rhizome. These branches perhaps helps in vegetative propagation. The tip of the aerial branch usually bear a solitary terminal sporangium, which was about 12 in length and 4 in height. So this is the external morphology of the Rhynia, where you in the figure A you will be finding the Rhynia major, and you can see the in the B figure where you see the Rhynia von Vaughan. Regarding the anatomy of Rhynia, if you see the transverse section of the aerial shoot and rhizome, the shoots and rhizomes are almost similar, and they can be differentiated into three parts: the epidermis, cortex, and the stele. The epidermis is the outermost surrounding layer with one cell thick and covered with thin cuticle with aerial shoots were interrupted at certain places by stomata, but stomata were absent in rhizome. Cortex was developed by epidermis is followed by cortex, differentiated into outer and inner cortex. Outer cortex is 1 to 4 cells thick, thin walled and without intercellular spaces, while inner cortex has large intercellular spaces and is and its cells have chloroplasts. It is thought that this was the chief photosynthetic region of the plant, and the endodermis and pericycle layers were absent. So this is the Rhynia internal organization, where you can see the outer epidermis in the middle cortex and the inner the stellar region. The epidermis single layer and outermost without any intercellular spaces, and you can see the cuticle beyond it. This epidermis at some places is having the stomata, and beneath the is the cortex, which is differentiated into outer and inner cortex, where the outer cortex is few layers. It is have it is parenchymatous, and inner wall is also parenchymatous with intercellular spaces, and the stele is the protostele. The center of the aerial shoot was occupied by stele. The stele is protostele, which is the haplostele. Xylem was made up of annular tracheids, and there was no sieve plates in phloem. Regarding the reproductive structure of the Rhynia, sporangia bonds singly on the apices of some aerial branches. Each sporangium oval or slightly cylindrical structure with little greater diameter than that of the aerial branch on which it is developed. It is 12, the sporangia is 12 long and 4 in breadth in Rhynia major and 4 long and 1 broad in the Rhynia guan. A longitudinal section of the sporangium shows it has 5 cells thick wall. The outermost layer was 1 cell thick cuticular as epidermis. It was followed by 3 cells thick middle layers of thin walled cells. The innermost layer was 1 cell thick tapetum. The wall was surrounding a spacious sporangial cavity which was without columna and contained large number of spores. The spores were of same size and measured up to 60 micron in diameter. Rhynia spores are homosporous. In many species, the sporangium contained tetrahedral tetrads of spores, which suggests that they were formed by reduction division and the plant bearing them represent the sporophytic general. There are no special mechanism for sporangium dehiscence. Liberation of the spores takes place by disintegration of the sporangial wall, and nothing definite about the gametophytic of the Rhynia is known. This is the structure of sporangia, where you can see the fossilized spore having the sporangial wall, spores which are present inside. These spores are spore tetrads which germinate into the gametophyte.